All living organisms share several key characteristics or functions: order, sensitivity or response to the environment; reproduction, adaptation, growth and development; regulation, homeostasis, energy processing, and evolution. Living things are highly organised parts of a hierarchy that includes atoms, molecules, organelles, cells, tissues, organs, and organ systems. Organisms, in turn, are grouped as populations, communities, ecosystems, and the biosphere. The great diversity of life today evolved from less diverse ancestral organisms over billions of years. Nucleic acids are the most important macromolecules for the continuity of life. They carry the genetic blueprint of a cell and carry instructions for the functioning of the cell. The two main types of nucleic acids are DNA, also known as deoxyribonucleic acid, and RNA, which is also known as ribonucleic acid. DNA is the genetic material found in all living organisms, ranging from single-cell bacteria to multicellular mammals. DNA has a double helix structure. The sugar and phosphate lie on the outside of the helix, forming the backbone of the DNA, whereas RNA is mainly involved in the process of protein synthesis under the direction of DNA. RNA is usually single-stranded and is made of ribonucleotides that are linked by phosphodiester bonds. A ribonucleotide in the RNA chain contains ribose, the pentose sugar, one of the 4 nitrogenous bases, A, U, G, and C, and the phosphate group. DNA is often referred to as the blueprint of life, and it was discovered that the code represents proteins that make up everything in our body. The genes that determine whether you have green eyes or how fast you metabolize food are the same in the cells of your eyes as your liver, even though these organs function quite differently. Although each cell shares the same genome and DNA sequence, each cell does not turn on or express the same set of genes. Each cell type needs a different set of proteins to perform its functions. Therefore, only a small subset of proteins is expressed in a cell. For the proteins to be expressed, the DNA must be transcribed into RNA, and the RNA must be translated into protein. In a given cell type, not all genes encoded in the DNA are transcribed into RNA or translated into protein because the specific cells in our body have specific functions. Specialised proteins that make up the eye, like the iris, lens and cornea, are only expressed in the eye, whereas the specialised proteins in the heart, the pacemaker cells, heart muscles and valves, are only expressed in the heart. At any given time, only a subset of all the genes encoded by a DNA are expressed and translated into proteins. The expression of specific genes is a highly regulated process with many levels and stages of control. This complexity ensures the proper expression in the proper cell at the proper time. Genetic variation happens through mutation, that is, random changes or copying errors in the DNA caused, for instance, by the exposure to chemicals and radiation. A gene may exist in many slightly different variants, called alleles. For a mutation to be passed on to the next generation, it has to happen in the sex cells, that is, the sperm or the egg. A QUT study into the DNA links for the migraine, a debilitating disorder. Researchers have identified several genes implicated in migraines and are translating that information in new diagnostics and treatments. One of the genes causes a mutation in the particular enzymes, which stops the enzyme from working properly. QUT trialled a specific combination of vitamins that made the enzyme work better and made the gene then function properly. Long before chromosomes were visualised under a microscope, the patterns of inheritance were first brought to us by the father of genetics himself, Gregor Mendel. Traits are inherited through the combination of maternal and paternal alleles. Each gene can be expressed differently as a result of its allele combination and mode of inheritance. The behaviours and interactions of chromosomes during meiosis explain, at a cellular level, the patterns of inheritance that we observe in populations. Genetic disorders involving alterations in chromosome number and structure may have dramatic effects and can prevent a fertilised egg from developing altogether. All life on Earth is related. Evolutionary theory states that humans, insects, plants and bacteria all share a common ancestor, but that millions of years of evolution shaped each of these organisms into the forms we see today. Scientists consider evolution a key concept to understanding life. Evolution is a process of adaptation through mutation, which allows for more desirable characteristics to be passed on to the next generation. Over time, organisms evolve more characteristics that are beneficial to their survival. For living organisms to adapt and change to the environmental pressures, genetic variation must be present. With genetic variation, individuals have differences in form and function that allow some to survive certain conditions better than others, these organisms pass their favourable traits to their offspring. Eventually, environments change and what was once considered a desirable, advantageous trait may become undesirable, and organisms may further evolve. Evolution may be convergent, with similar traits evolving in multiple species, or divergent, with diverse traits evolving in multiple species that come from a common ancestor. Evidence of evolution can be observed by means of DNA code and the fossil record, and also by the existence of homologous and vesicle structures. Natural selection, also known as survival of the fittest, is the more prolific reproduction of individuals with favourable traits that survive environmental change because of those traits. This leads to evolutionary change. Natural selection acts to promote traits and behaviours that increase an organism's chance of survival and reproduction, whilst eliminating those traits and behaviours that are to the organism's detriment. Improvements in gene pools come from things like mutations or immigration of species to build and develop more favourable traits for survival. For example, QUT have been able to trace the body size change over the evolutionary history of kangaroos and wallabies. With DNA sequenced from inner ear bones of a 45,000-year-old giant short-faced kangaroo, scientists have been able to confirm that the kangaroo and wallaby ancestors diverged from a tree-living possum around 40 million years ago. They remain small, in the 2 to 15 kilo range, while Australia was more dominated in rainforests. And as the climate cooled and dried, and as forests opened up over the past 10 million years, at least 4 different kangaroo lineages independently evolved to megafaunal size, more than 44 kilos. Although each time a species diverges through evolution, patterns can be seen and clues are left behind for scientists to discover hundreds of years later. These dominant traits being inherited and passed on leads to the extinction of species that are not as well adapted, and although we cannot see these species face to face in our modern times, technology helps us recreate the idea that these creatures once walked the same earth as us. Millions of years of evolution and natural selection have turned the biological world into an effective materials development laboratory. Biotechnology involves the use of living systems and organisms to develop and make products. Biotechnology research can help reduce threats to food, health and water resources and minimise the impacts of climate change, crime and terrorism. QUT is leading efforts to create a super banana that is disease and drought tolerant and genetically modified for increased nutritional value. These bananas have the potential to save millions of people from starvation in underdeveloped countries. Taking inspiration from the natural world, QUT scientists are focusing on using structures found in the natural world and applying them to existing materials to try and discover new properties that can then be used for technological advances, such as using the nanostructures of fly eyes to develop anti-fogging coatings for extreme weathers and copying the structure of honeycomb to build better lithium-ion batteries.